Hello, in this video we will discuss about the Luciferase reporter gene assay. What is this? Let's begin to understand the whole process and the Luciferase reporter gene assay, their application. It is used for identification of gene function and disease identification. So how it is possible? Let's begin to understand. This is the native cell, that is normal person cell, which that contain promoter and natural gene. Promoter is responsible for the promote the natural gene to activation to produce messenger RNA is known as the transcription. After the messenger RNA production, it is known as the transcription. The messenger RNA will go into the cytoplasm to produce protein when bind with the ribosome is known as translation. And this protein is functional after the modification in the endoplasmic reticulum and Golgi bodies to produce this protein is a functional protein, and this is the endogenous gene product. So let's begin to understand this promoter after the sampling of the cell, the tissue, which they are sampling to segmentation, the fragmentation via endonuclease enzyme to produce the amplification by the putting the primer, specific primer for the promoter amplification in the nucleotide buffer and sample which that put in the PCR for amplification of the promoter of interest. After the promoter of interest amplification by the specific primer binding the denaturation to annealing to extension cycle which that produce the promoter specific, and this is the promoter of interest. This promoter of interest will insert by a ligase enzyme and restriction enzyme into the plasmid. After the binding with the plasmid, also the luciferase producing gene, which that is our also interest, but this is the general a gene we can add, and this is the 3' untranslated region microRNA recognition element MRES, which that is specific cell which that will lead to inhibit this function, but the that cell which that will not inhibit the luciferase producing gene, that means that is our choice of that cell. So how to check the function of cell by reporting gene assay? First of all, we will learn here and what is the function. So we will choose the transfecting cell as a transfection. So the transfection mean the basically is the transfection mean the cell which that this plasmid will insert into the cell is known as transfection, and the cell is known as the transfecting cell. And after this transfecting cell recombinant introducing cell, the transfecting cell is known as recombinant cell. And this after the introduction of the plasmid via electroporation, lipid micelles, and calcium phosphate, for example, which that contain positive charge is helping to the negative charge containing DNA plasmid will insert. After the insertion, the recombinant introducing cell transfecting cell which that will produce the color after the luciferase activation to luciferin to oxyluciferin, basically is the so a particular time required for the production of the color is known as the bioluminescence. So anyhow, let's begin to understand the how to check the strength of the promoter. We will understand, but it is used for identification of gene. Remember again, function and disease, the of gene function and disease, and reporter gene identification of signaling pathway working for cell. So remember the reporting gene identification of signaling pathway working. So the reporter gene identification of strength signaling pathway working for cell. Sorry. So the signaling pathway working. So how it is work? Let's begin to understand and how to check the strength of the promoter. Then we will learn from the native cell at different situation. So the native cell means that cell which that are present in our body, that in the form of tissue, bones, or the brain cells or other type of cell, which that function differently, and the signal required as a ligand will bind with the receptor. After the binding with the receptor, it will change the physiology, that change the cellular pathway, the function of the cell will be changed. So how it is possible? For example, the normal cell is here, and this is the abnormal cell, and this is the non-functional cell, for example. So when the ligand will bind, it will, the cascade of phosphorylation will occur, will target to the promoter, and when target to the promoter, it will lead to trigger the gene, the natural gene will produce product. This product if more, so it means the basically the promoter is strong, while the abnormal mean the product will be less, it means weak, while the non-functional mean no product will produce. So here is after the removing of the promoter will fix with the luciferase gene in the ex vivo. So in this way here you can see the non-product basically is the non-functional if the product will not produce in the laboratory, and the promoter produce a lot more luciferase, produce more color, is it means more strong, it means it is functional more, and promoter weak when product less than strong, so it means we can identify in the cell by this mechanism. So that is why the genetically more beneficial this luciferase reporter gene assay. Anyhow, let's begin to understand gene expression in specific cell. So the specific cell requires the specific promoter. So in this way identification we will learn from this. So after this promoter requires the specific cell for the promotion of the gene function. So for example, when we will insert this plasmid, recombinant plasmid containing reporter of interest and the reporter like GFP or luciferase. In this way the luciferase will produce reporter protein, luciferase produce reporter protein, D-luciferin will convert into basically with the oxygen and ATP to the oxyluciferin, and carbon dioxide produce AMP, and light will produce. So in this way, I hope you make sense about this. Luciferase enzyme is used for the production of light when the luciferin are present. So that is why. For example, it will insert into the different cells. For example, the epithelial cell, stem cell, fibroblast cell, and neuron. So for example, if neuron will not work, so it means the promoter of interest is not specific for the neuron, because the neuron requires the specific promoter. It means we can identify this promoter is used for only the epithelial cell specific. So the epithelial cell and stem cell specific, for example, when produce the color. So that is why we can say that when we will add the luciferin protein also, or protein are already present, so it will convert into the oxyluciferin in the presence of luciferase enzyme. So remember, and the epithelial cell and stem cell is functional, so we can say that the epithelial cell specific promoter this is. So let's begin to understand the normal ratio calculation. The normal ratio calculation means the normalized ratio is equal to experimental reporter activity divided by the control reporter activity. So the control reporter activity. This method accounts for well-to-well variability caused by differences in we will understand. So here is the experimental reporter activity divided by control reporter activity, which that is equal to normalized ratio. How to calculate the normalized ratio? By this mechanism, by this equation. So in this way that this method accounts for well-to-well variability. So well-to-well variability, we can calculate the average and normalized ratio by differences in number one is the transfection efficiency, Number 2 is the cell number of viable cell, or the 3 is the edge effect caused by position on the plate. So let's begin to understand here with the experiment. So this is a normal ratio calculation. We can understand where this normalized ratio is the average normalized ratio, and divided by the average normalized ratio for what? Let's begin to understand, and we will put the number. So for example, we are checking the drug activity. So the promoter is required the drug for production of protein due to the drug. So for example, sample A and sample B, X and X1, which that contain a drug efficiency checking. So in this way, the luciferase protein produce luciferase enzyme when produced in the sample A will more less, while the sample B will more. In this way, you can say that the sample B is the drug X1 is producing more luciferase. It means the promoter is strong for that drug, while the promoter is less strong in sample A for that drug. So it can be divided by the sample A in X and the sample B divided by sample B X1 to normalized ratio calculation to understand by this valve. So this valve containing many sample to show the color, and we can calculate and normalize the ratio to understand. So basically is the luciferase reporter gene assay used for the disease detection and gene identification for the working, as well as the defective gene understanding. On the other hand, the non-product producing non-functional, less functional gene identification. So thanks for watching. This was a video.